Probably. Okay, sweet. Hey everybody. So yeah, a little more about myself. I'm a huge nerd. I love this beautiful, earth we get to experience. And yeah, have kind of like a deep background with farming, like growing food, soil, water, environmental health, also mushroom, like farming, cultivation for food and medicine. And then really though my focus and biggest passion has been working with fungi to clean up, environmental pollution in all kinds of different contexts. But I actually work with mycorrhizal fungi a lot in mycoremediation as well. Although that's just one application. So really, mycorrhizal fungi are fungi that live in symbiotic, mutually beneficial relationships through the roots of plants. And most plants on Earth, like greater than 95% can form these relationships. So you're talking about cacti, basically especially like more extreme environments like deserts, ecosystems, pretty much every plant is going to have a mycorrhizal partner. They would not be able to really survive in those rough environments without them. But they're also found in wetlands. There's like aquatic mycorrhizal fungi. There's mycorrhizal fungi in the boreal forests and pretty much everywhere. So I thought we'd start with just a bit of a recap of part one, and then talk about what we're gonna do today. But yeah, so in part one, we gave more of the background on like what are mycorrhizal fungi, what are the different types. There's a whole bunch of different types. Does anyone remember one or want to call out like one type of mycorrhizal fungus? Yeah, cool, ectomycorrhizae. And anyone know what kind of plants ectomycorrhizal fungi usually connect with? Pines. So a lot of coniferous trees and like long-lived trees. So ectomycorrhizae, they're like a type of specialist mycorrhizal fungi. So they form associations with like 3 to 5% of plants globally. It's just they have a really big spread because pine trees are found pretty much everywhere in the world, right? There's just different kinds. But so anyone know the generalist mycorrhizae, the ones that form relationships with like 90% of all plants, including crops, fruit trees, even like oak trees, and a lot of like, yeah, good sleuth. Nice. No, good call. Yeah, yeah. So our bus. No, it's all good. Yeah, you're paying attention. It's great. Yeah. So Arbus, anyone speak French here? Yeah. Yeah. What is it? Arbuscule sound like? But it's yeah, tree. Yeah. And so we're gonna get to look at this firsthand. But if you take a look, these little marketplaces where the arbuscular mycorrhizal fungus actually goes into the root and trades, nutrients, water, all kinds of stuff with the plant, are called arbuscules. And they're called that because they kind of look like little trees. So yeah, those are the two main categories of mycorrhizal fungi. There's a whole bunch of other types. There's mycorrhizae that form relationships with orchids, and orchids can't germinate like without these mycorrhizal fungi. So there's ericourt, there's a whole bunch of other types, but these are the two main ones that form relationships with most plants on Earth. But just as kind of a recap, if you're working with specific plant types, it's good to figure out what type of mycorrhizal association they form, right? So usually, pretty much, again, most plants will form relationships with AMF, the arbuscular mycorrhizal fungi. Even the ones that end up being ectomycorrhizal later in their lives, often when they're young, they get their start with AMF and then transition to EMF over time. So, yeah, so, okay, two different types. the main thing, and I just wanted to recap this because we're going to be looking at some roots and hoping to find these different mycorrhizae. Major differences between these two: the ectos just form a sheath, like, so you can see this kind of yellowish white stuff. This is a grape root, I think, all around the outside of the root. That's the ecto. It like you can see in this kind of cross section of a root, it's just gonna build up its hyphae basically around the root, and then go like circle the cells to trade that way. Whereas again, the AMF is going to actually penetrate root cells and trade through those arbuscules. Yeah.
How far out into the environment
Like what type of coverage globally,
yeah,
or
so you've got it,
yeah.
for the ecto, it's right around the root. Does it also go out 3 feet?
Yeah, in healthy undisturbed ecosystems, these networks would be essentially continuous, like everywhere. Deserts, grasslands, forests, wetlands, everywhere except for like the ocean, you know. And they're dynamic, yeah. And so they're connecting. I don't have a picture of that. There's some cool visualizations of what these networks look like, but you might have heard about like mycelium being kind of like the true internet globally. They're talking about, when they're talking about the wood wide web, they're talking about mycorrhizal fungi. And so these networks will include like so many different types of, you'll have AMF, EMF, all kind of woven together, connecting with different plants and connecting all the plants in a system. And just what disturbs them, like building roads, cutting down their like cutting down forests, industrial agriculture, with where you're applying a lot of fertilizer, right? So the main trade going on, I mean, these fungi are trading a lot of different things with their plant partner, but one of the major trades is phosphorus, which can be present in soils, but not available to the plants. Even if it's there, it might not be available, right? So these fungi are really good at basically gathering up phosphorus, making it available to the plant, and trading it with them. And if we're fertilizing and just giving the plant that fertilizer, it's not gonna maybe invest in this underground relationship, which has so many other benefits. I mean, generally, plants grow so much better when they have their mycorrhizal partner, especially in the face of stress. So whether it's like, temperature extremes, drought, flooding, like whatever, heavy metal stress, whatever the type of stress is, these underground fungi are going to help protect the plant. It's their plant partner. Okay, so yeah, we went through just recapping some of the major types. Part one, which we have a handout for, we can share an email, basically went through how to cultivate these yourself. And so why would you want to do that? Because you can just buy, inoculum that's been produced. But the thing is, those inoculum, they're only really produced in like a couple parts of the country. And so there's huge benefits to having a bioregional inoculum, where you're literally what we did is we went to find old trees of the 3 different tree species that they're working with in this program. We scooped up soil from the root zone that should have these mycorrhizal propagules, and then we basically cultivated them or expanded them in a really simple way. So this is different though. So for mushroom cultivators, like, how you have to figure out what the right substrate is for the mushroom you want to grow, right? So it's different with mycorrhizae because they only get their food from their plant partner. So they cannot live without a plant partner. So if you want to grow them, you have to plant you're not like, you have to plant a plant species that they will form a relationship with, let that relationship form over a period of a few months, and then basically let the plant end its life cycle. So often people will use grasses for for mycorrhizal cultivation, you basically like stop watering and then cut down the plant. So basically what that's telling, remember this is it's called obligate symbiont, meaning like these mycorrhizae will only live if they have a plant partner. So once its plant partner is dying, it's like, oh, I gotta reproduce. It's gonna focus on, it's gonna shift from, it's like, it's probably gonna try to help the plant, be like, do you need more water? Do you need more nutrient? What do you need? You know, but once it knows that the plant is dying, it's gonna put spores out into the soil and try to continue its life cycle at a later time, right? Does that make sense? Yeah. So basically what you're coming into today is last time, about 3 months ago or so, we set up all those um, pots of basically a mix of sand and coir, that we literally just added like handfuls of the soil we collected from the root zones of those native trees, and which is our inoculum, right? So that should have had like spores and other mycorrhizal propagules in that soil. And then we planted, what was the grass again? So we used ryegrass and basically let it grow, water normally as if you're, trying to grow these plants. And then a couple of weeks ago, stopped watering and cut down the grass. So that would have signaled to the mycorrhizae it's time to put spores out into the soil. So um, what we're going to do today is not a necessary step. So like, well, we'll show you how you would use this as inoculum, because technically right now this is already an inoculum. You would, you can like shake the soil off, chop up the root bits. So all the root bits that are infected, it's weird to call it infected, it's positive. But yeah, you can, the root bits, yeah, which is also a weird term. So yeah, I know. So yeah, you chop up the root bits, all the soil in here. This is now your inoculum. So you can literally, you can store it in these pots in a cool, dry place for up to a year for use. If you want to store it longer, you can basically put it in some Ziplocs and store it in the fridge. But you go from like, we collected, I think, like a couple buckets worth of soil from these, root zones of these different native trees, and we end up expanding it. So in that way, it's the same as mushroom cultivation, right? You're just like starting with a little bit of inoculum, you give it more food and the right conditions, and you end up with a bunch more. So again, why you're like, oh, you could still, why did you not just buy it? It's so much easier. It is, but there's like been multiple meta-analyses where they looked at all the available studies into inoculation that have been carried out, and they tend to find that inoculation with commercial mycorrhizal inoculum is not effective, meaning it doesn't work. Like, so they go back and do what we're doing, which is a way of checking, like, hey, did this work? Did we end up with the mycorrhizae we were hoping to propagate? And there's a lot of reasons that may be. It could be because they're not from the region and they're just not adapted to these specific conditions. It could be because these guys they help their plant partners be resilient, but they can be a little sensitive, which is also why I like to teach people how to grow them and protect them. If you've heard of a cool organization to check out is SPUN, Society for the Protection of Underground Networks. Have you heard of them? Yes. But so they're all about learning, like basically through mostly they use sequencing tools to try to learn about mycorrhizal networks in understudied kind of regions around the world, because they're saying like these essential fungi are being damaged and destroyed as we are damaging and destroying huge areas of whole ecosystems, and we need to like learn about them and advocate for them to be protected. So commercial inoculum don't really work very well. A lot of reasons why that might be. Yeah.
So does that mean mushroom blocks to try and remediate soil is kind of a pointless endeavor?
No, no, it's a totally different thing. So usually when I'm doing remediation, I will work with decomposer fungi, which are the blocks, and they get their food by breaking down dead stuff, carbon. So they can also break down organic contaminants, which are carbon based, so like petrochemicals and stuff. Mycorrhizal fungi, can anyone get, are you saw my how do you think mycorrhizal fungi can help with remediation? Think about the role they play and what they trade with plant partners. 100, yeah, totally. So if you're working with plants for remediation, then these guys are going to help those plants survive in toxic soils, right? Another thing I've found in my own research is that arbusticular mycorrhizal fungi help their plant partner take up more of the metals of concern. And that's a way, because metals don't break down, you need to remove them into plants. So like lead is an example of a metal. Um, and yeah, these AMF really can help with that. So um, yeah, okay, so commercial inoculum doesn't really work. But also, have any of you been involved with like large scale ecological restoration at all? Wanting to be. So finally people I don't know, it's kind of blows my mind, but for a long time ecological restoration efforts would not consider the below ground networks. So they would just plant trees, plant, you know, whatever plants, and they would like all die for real, like greater than 90% mortality of what they planted, and they just keep planting and planting, and finally people were like, oh, it's because these plants need their, not just mycorrhizal partners, but I'm sure it's a huge piece. They need all the microbes that help them grow, especially in an ecosystem that's degraded. But the major way, because ecologists don't love commercial inoculum because they're not regional, you know, they're not native, and they don't work very well. Um, Ecological restoration started using what's called a whole soil inoculum. So they would literally dig up an entire undisturbed ecosystem, like huge quantities of soil, and use that to inoculate. Like basically, it's like a microbiome, soil microbiome transplant for the area they're trying to restore. But in doing so, you're like, what is that saying? Like stealing from Peter to pay Paul? Yeah, it's like you're like destroying one ecosystem to help another one heal. And so this method is great because you're essentially using that same principle, right? You're still scooping, but you're only taking like a little bit of soil from that healthy ecosystem, and then you're bulking it out and expanding it. And now you have your huge amount of inoculum, which is going to have not only the mycorrhizal fungi, but probably other microbes in the soil locally that you can now use in remediation projects, restoration projects, agricultural projects, um, really anything involving plants.
For with these pots, how much of the root soil put in each pot?
You wanted we did like a handful or two. You're supposed to have a ratio of like um, essentially 1 part like whole soil inoculum to 4 parts of your potting mix. Um, yeah, and so basically what our plan is for today. Any more questions though before I move on? Yeah.
How do you know if you already have the mycorrhizal in your land?
Yay! That's exactly what we're doing today. So perfect segue. So the methods I'm going to show today are ways to evaluate whether you have mycorrhizal fungi in your soil and whether it is successfully forming relationships with your plants. So you could do one or both of these or none of them at all. Like if you were just trying to propagate, if it was me, I would want to just check to make sure you ended up with, you know, successful mycorrhizal propagation. And you can use these methods. Um, and you can do that in your initial soil that you collect to be like, okay, is this a good inoculum to use to bulk out? But you can also just do this if you're curious about whether your plants are forming mycorrhizal relationships. I like to do this when I move to it or when I'm in a new ecosystem. It's just a cool way of like learning about underground networks that aren't visible to us. And so I do want to say too, the methods we're showing today are often thought of as like high-tech methods where you need like a lab, a bunch of equipment, a expensive microscope and things like that. But it isn't the case. I've done this like in, remote setting. I was like where I'm hiking around collecting samples and then using a $10 microscope and a lot of reagents, I'll say, you can get from like the drugstore. So there's ways that I've adapted these methods that are often carried out in like, a graduate program or like a university, to be a little more doable by folks who maybe are like just wanting to learn more and try this stuff out. And yeah, we can email you the methods with the adaptations I've added. But today the plan is we're gonna just literally do it. So there might be, I've tried to set it up so that there isn't too much like sitting around time. But these methods just take time. Like sometimes we'll have to be like letting something sit for 10 minutes and so on. So I'm gonna walk you through all of it. There's a bathroom right here and then more back there if you need. We should probably we'll take a break at some point too. But yeah, basically I'm gonna show you spore extraction and what's called root staining, to look for mycorrhizal fungi. And then we'll do some looking under a microscope and I'll show you how to identify like, what are you seeing? Did it work? Did it not work? Um, and then, yeah, if there's time, and we have good mycorrhizae going on in our samples, maybe people can help process some of the inoculum we produced in the last workshop. Yeah, so that's the plan. It's a real methods. I mean, these have both been very methods workshops, but this one is very like kind of like DIY lab methods workshop. Okay, and because there's so many detailed like timing pieces, I have detailed instructions. So, okay. Can you remind me? Yeah. Cool. Okay. Does anyone remember or know, like, how do you think that mycorrhizae help with drought tolerance? We know they do. Any guesses how? Yeah. Yeah, totally. So I did my PhD in Southern California, and in the deserts, you'll have stands of oak, like in just like pure desert. And the reason those oaks are able to live in the desert like that is because of mycorrhizal fungi that literally grow like 30 to 50 feet through the sand, penetrate the bedrock to access the groundwater and aquifers, and pull up that water and send it up to the tree partners. So there's like wild stuff going on down there. Doesn't it also have like a coat, like keeps the water inside? Yes. Yeah, you got it. That's the second piece. I think I have a picture. So AMF produced something called glomalin, which is this sticky stuff that I had a picture at some point. It's this sticky stuff that its whole purpose why they make it is to hold moisture around the root zone of plants. But glomalin is also cool because it's thought of as this like underground vault for carbon. So apparently just glomalin through mycorrhizal fungi sequesters like a third of all carbon in soil, which is huge. Okay, cool. So our first step is we're gonna get our samples ready, harvest our inoculum. So basically, I'm just gonna shake off the soil into the bucket. I'm just gonna try to like separate the roots off.
At this point, do you know you were successful?
No, no, we haven't. This is us checking with you in real time. Yeah. Okay, so basically, yeah, so I've got the soil with all the spores in it, hopefully in the bucket. And then I'm gonna use a shovel, or you can use like a, an axe or something to basically chop up the roots, because the roots will also contain propagules. Is this like moist or? No, it's dry, and that's because we intentionally stopped watering a few weeks ago so that the plant would die and signal to the
Get close to the spore.
It's a good question. AMF do not produce any fruiting bodies ever. Ectomycorrhizae do, and so most of the mushrooms you'd harvest in the forest, like from matsutake mushroom, pine mushrooms, they're called that because they're mycorrhizal with pine trees. Chanterelles, a whole bunch of different mushrooms you'd pick are fruiting from ectomycorrhizal networks. They need some time. Okay. Yeah, so there might be there should be spores in the soil if it's been living there a while, but you're not going to have as much as if it has a bit of time to be like, oh, I got to start reproducing. Yeah. Kind of like if you thinking of a cultivation analogy, kind of like if you like somehow fruited prior to the fungus having grown through its substrate, it might produce a few little ones, but it's not going to be the full amount. Cool. All right, and then I'm just gonna try to like get the grass out. We don't need that.
Was your effort purposeful for the time of year?
That's a really good question. It is, I mean, so you can time this with the seasons. So like, I mean, with this, we kind of did, right? Yeah, we like, so for example, that they die back on their own because of seasonal, like it's getting hot or whatever, so you can definitely time it that way. It would be harder to do. I mean, you just need to pick also a plant symbiont that will be suited to the season you're growing. Yeah.
And we're gonna do another round of growing sorghum, a different type of grass, to grow more of the inoculum. So we're using kind of the seasonality of the plants to be able to have two, like throughout this year. So we're using them for our plantings in the winter of 2026, 2025, 2026. And so we'll be growing sorghum here in another couple of weeks. We'll be planting it and potting it up, doing basically the same thing that we did with the ryegrass this winter.
Cool. Does anyone want to help? Could have a few helpers. So we need one thing. It looks like you want to help. Do you want to get about 5 grams of soil? We can you can kind of sieve it if you want.
5
Yeah. Or sorry, 5 milliliters.
Oh, 5 milliliters.
Yeah, fill to the 5. Yeah, sorry. Wrong units. And try to avoid like the roots if you can and just get the soil. So this is for the spore extraction part. So I'll try to say like this is for spore extraction or this is for root staining because we are going to be like doing both at the same time. Root staining. So root staining will hopefully give us an ability to look at different mycorrhizal structures inside the roots like you're seeing here. Spore extractions will show us the number of spores and types. You can actually identify down to genus and even species if you get comfy with like how to look for different spore types.
The collections you took
Yeah.
require the same host plant. It's okay to use the grass.
Yeah, totally. Yeah.
So those guys are just opportunities. They like
Yeah, the AMF are generalists, so they form as long don't plant brassicas. They don't form relationships with brassicas, and yeah, other than that, pretty much any plant that will work. Yeah, we have petri dishes too. Yeah, which. Oh yeah, or here's an idea. How about that?
All of our tools.
Yeah, which is actually good to show because again, I'm trying to show like you don't need to have all the things. You can improvise. I'll try to be clear what things are really important and why. Nice. Sweet. Cool. Great.
Good job.
Nice. Does anyone else want to grab, or you can definitely fill another one? Someone else? Yeah. Love it.
This works pretty well.
Oh, you can fill it. I thought you needed to take the top.
Can I hand this to someone? Does someone want to do the next part? Okay, cool. Yeah. So
Tree Folks is going to pair, or you're going to pair these spores with their tree companions when you plant. Yeah, we actually have
oh my gosh.
commercial study.
So if you fill it up to the 14 mil mark, please. Cool.
So we're going to see how that goes, and then we're going to read.
No. So once you have your soil sample, you basically just fill it up with water, and then you're gonna shake it.
Getting that slurry. Oh, I'll take some shots after this.
Shake it a lot. Here we go. Okay, there you go. You have the lids there. Thank you. So do you want to shake this up thoroughly? This is just water. Oh, cool. It is distilled water, but it doesn't matter too much for this. So shake. Sorry, which?
The HB level doesn't matter and stuff like that.
No, okay. Not for this. Yeah, and so we're just shaking it really thoroughly. You want it to be like all mixed up, and then we're gonna once it's all shook up, we're gonna lay them on their side. Actually, April, could you hand me my phone, please? We're just gonna lay them on their side for 15 minutes. So I'm gonna set a timer.
timer set?
Yeah. Okay, cool. All right. Any questions on this before we move on to the next step? Yeah.
Oh, she was asking about how to store the spores in the inoculum after.
Yeah, so if you're, I mean, for the bulk inoculum, you can just store it in a bucket in a cool, dark place for up to a year. If you want it to last longer, you can store it in like Ziploc bags in the fridge to help it last a little longer.
Is there a temperature? Because Texas can be
I know.
Is there a temperature of what mycorrhizal can know, like goes kind of bad?
Yeah, but it's really hot. It's like I think it's like 125 or something.
Which can happen in Texas.
Which can happen. So yeah, no, good point. Like, so I think if you really want it to last for sure, if you have like do people still have like cold cellars?
Not really.
Not really. Okay, you don't have it. Okay. Yeah. Well, basically, then what I would recommend is just to time it with when you want to plant. You know what I mean? So like, do your production. So do your production and then just be ready to use it as inoculum or keep it in buckets. Yeah.
How sensitive is a like live mycorrhizal, like to being disturbed? Like saying you want to transplant something and that you know that there's good live mycorrhizal in the soil, but it needs a bigger pot. So you need some more fresh soil, but need to disturb the soil. Will it easily continue to grow with the new stuff?
Yeah, for sure. I mean, if you're like tilling will definitely disrupt the networks. But short of that, yeah, if you're just like digging it up, it should be okay. Yeah. Yeah, for mycorrhizae. Yeah, basically all industrial agricultural practices are no for mycorrhizae, which is why you might want to restore their populations. So usually they should be everywhere. The places they sometimes aren't are like industrial agricultural sites, super contaminated, degraded sites. They'll be there, but in really low concentrations or abundances. And like so sites like that, even like post volcanoes and stuff, there's all this cool research one of my mentors did about how they repopulate. They repopulate slowly. Their spores are actually big, even though we can't see them with our naked eye. They're large, and so ants will carry them, gophers carry them, all kinds of like anything that is going to be walking around and living in the soil will actually carry them to new locations and help them kind of repopulate. But the problem we have in today's world is that, we've taken away those kind of mother networks for them to repopulate from. So if we have like, an urban area, it's going to be, they move very slowly. If an ant is carrying like one spore, however far, right? If it's like really far to a forest or undisturbed ecosystem, it's just going to be slow and difficult for them to repopulate. So you might want to, you could check first, see if they're there. And if not, you might want to do something like this. Yeah.
Can you like throw your inoculum in the fridge to keep it
Yeah.
for a while?
Oh, for sure. Yeah. Yeah. And in the pot, like this is a pot grown in mostly sand. And so sand actually preserves the spores really well. So like we talked about this a bit last time, but if you're like want to make a business doing like regional mycorrhizal inoculum or something, you would not, well, you would do all of this, except you would probably like extract the spores and literally store them in sand in the fridge and like have them ID'd, and you'd have like, that would be like having a, like your culture essentially, that you're like, I know what this is. It's a blend of like glomus, blah, blah, blah. And this.
Would freezing damage?
Yeah, probably. Actually, that's interesting. Yeah, I don't know, but I think so. I think so. Yeah. Yeah, the spore is its like propagate, like its reproductive kind of piece. So they can be resilient, but not to like temperature extremes. That's a good question, though.
Is the sand because of more porous?
Sand is just a good preservant. Okay. Like my grandma used to they'd bury their crops in sand in the cellar to preserve them because it can stay dry. So yeah. Okay, cool. So our next thing we're gonna do is switching over to getting our roots prepped for root staining. And so all we're gonna do to start, and so this is the only kind of specialty piece of, I guess, like material for this. These are used in dentistry. I don't know for what, but they're called histocassettes. Yeah, histocassettes. You can order them online, but if you want, I would just coordinate because you can only buy them in like sets of like 500. So probably I'm sure they have enough to, not to like offer that, but like maybe we could do it as a community if you want to do this stuff. So these are called histocassettes. What's awesome about them is they're just this little cage to put your roots in, and they're like they can withstand chemicals and heat and stuff because we're going to be basically clearing the roots. We want to get not only all the soil off, but we want to clear all the stuff, and then we want to acidify them so that's because if you do dye stuff, you have to do a step to basically make the material ready to take in the stain or the dye. So then we acidify them, and then we basically stain them, and then we kind of de-stain them a bit, and then we can look at them under the microscope. But this is just a really easy way to get our little roots packed in and in a little cage that we can easily.
Does it have holes in it?
Yeah. Yeah.
Oh, okay.
So it allows the penetration of like all the chemicals and stuff. Yeah, and so basically I'll just show how this works. So first thing is I'm gonna get a nice root sample in this sieve, and then I'm gonna just wash it with some water to try to get some of the soil off to start. This is easier if you're just like have a sink, but definitely doable kind of in the field as well. Yes, please, thank you. Totally. Yeah, just trying to get, and we'll do some steps to clear these even more, but it's pretty important to get this stuff off because otherwise it just is hard to see what's going on under the microscope, and it might make the stain not work as well. And if you have woody roots, you need to do these processes for longer, just because it'll take a little more to get them fully cleared. Okay, so that's decent sample. I'm just gonna take a little bit of this and pack it in this. Actually, do you mind grabbing more of the cassettes, please? I think they're just on the table there. So we'll make a few of these. Thank you. That's pretty good. April, do you want to make one? Sure. There we go. Does anyone else want to make one?
Are we just putting it in? Or
yeah. Yeah. Oh no, do you want to do one? There you go. Oh, and then we'll just do one more. Do you want to? Okay. And then you can just like snap it closed. There we go. Okay. Thank you. Okay. Now, April, can I have you get our little stove going, please? Thank you. Okay, cool. And if you're doing this in like if you're doing a full study and you're going to be doing a lot of these, and you have different samples, you can use pencil to write what your sample is. Like maybe you're like, oh, I looked at the tomato, pine, whatever. Marker will come off in all the chemicals, but pencil won't.
Two sides or one side?
Just one side. Yeah, one side should be great.
All right, thank you so much. So it slides a little bit.
Okay, cool.
So there you go.
And if, woo, Wendy. So the method actually calls for KOH, which is like a low-key intense chemical that you definitely can get off the internet, but it creates like a toxic waste thing and blah blah. So we've replaced it instead with NaOH, which is also, like, as a heads up, like if you are working with NaOH, it makes an exothermic reaction when exposed to water. So that means it creates heat. So please be careful. Don't mix it and then like grab the jar or something. It will be hot. Okay, so basically, thank you, April, for preparing these solutions. Oh, NaOH is available as lye. It's also called lye. So you can get it from, drugstores. Where else can you, 100, yes, it's used in soap making. So you can get it pretty easily. And
something
which side is?
Over the
Yeah.
right side is.
Okay.
Over and over again is always add the NaOH to water, not
Yes.
the other way around.
Cool. So basically, now I got to set another timer here. So we're going to do this for 20 minutes. Oh, I already have one like that. And so like with any recipe, right? There might be slight variations based on the type of plant, but the signal you're looking for, this step is all about clearing the roots, right? We want them to be like not brown, basically, like just cleared so that they can hold the dye. So you can literally see a color change. You have a clear liquid that you're putting it in. When it's done, around 20 minutes, it should be like, you'll see the water should be, yellow brown, and be cleared. And you can already see like some of the dirt and stuff is coming off in there. Okay, so got my timer set on that. We're 1 minute away on these guys. Cool. Is it on the lowest setting? Let me see.
It's low. What do you want?
Okay, no, that's good.
Turn it
to okay.
So it's on the right side, I think.
Okay. Okay, cool. Okay.
So do you want it on a low setting or a high setting?
You want it low. You don't want it to boil.
So would something like an instant pot work? Would you put it inside?
Yeah. Yeah. Like in a lab setting, you'd use a hot plate. So you can use anything like that. No, that would like legit explode, I think. Yeah.
That's a good question.
Yeah. 90C. 90C is what you're going for. Oh, okay. So because we don't have power everywhere here, our next step on our spore extractions, so they've basically like rehydrated, is we're going to centrifuge them for 10 minutes at 2,500 RPMs. So yeah, you want to do it? Let's do it. Okay.
Here we go.
So also like super exciting if you're, I mean, obviously the centrifuge is maybe one of the most, like one of the pieces of equipment most people don't have access to. But these you can get centrifuges for like $35 now, which is, so we got a little small one here. There's also, you can do like DIY, like drill centrifuge, but it's a pain. It's really nice. Has anyone ever done that out of curiosity? Like DIY centrifuge? So basically, the reason and this is cool. So soil sciencey people. So basically the purpose of this is to remove any organic matter. And you can see organic matter is lighter. It's going to float on top. And what we want is just the soil itself concentrated in the bottom. So we're gonna, do we have our plug-in situation? Okay, cool. Do you mind getting that? Okay, so I'm gonna set it for 2,500. I think it's already at that. Perfect. And then it was for, sorry, 10 minutes.
This is fine. Let's see it now. Does this work?
And if you're using a centrifuge, you always want to balance it, which means like you don't want to have like 2 on one side and nothing on the other side, because it'll go ka-thunk, ka-thunk, ka-thunk. Yeah, let's see if this works on this power. So let's see.
If not, it works on the job.
Okay, cool. Where's the...
You got the centrifuge working before.
There it is.
There you go. You got it. Wonderful.
Whoa, it feels like that lid is not gonna hold on.
Here, I'll just put my hand on it.
Really?
For 20
but we
minutes.
can't. Yeah. Doesn't I have? That's so weird. Oh, let's see.
I have like, okay, let's get the deck.
Oh yeah,
it's
that's
dripping.
So that's why it's $35 a day. Okay.
Yeah, it's these tubes. It's
all
these
right,
caps.
all right. Did it come with tubes? No.
I thought it. Yeah, I thought it was going to. But,
okay.
You want duct tape, and then it can just strip down the tops of the. And at
I
some point, it'll just go
guess.
smooth. Because all the plastic will be.
Yeah.
It won't open them. It'll be. Music. It's a boy, Roger. Tricks. It's our next workshop. Another use a rope.
Okay, cool. Now
Yeah, look at that.
it's going. Heck yeah, love it. Okay.
Wow.
Yay. So, and this timer is set. Perfect. So 10 minutes. Cool.
Salad spinner would work.
You can. I have seen that. Yes, you basically tape them on the side.
Oh, yeah.
Yes.
2 young ladies from Rice won this enormous prize for bringing that to third world countries so you could centrifuge blood. Because you can do it hematocrit.
I love that.
So much.
That is so
Welcome
cool.
to Texas.
I love that. Okay, this is so great.
You know about that.
Thank you. Yeah, I forgot about that.
I know, there you go.
Yes. Yeah. Oh my gosh, yeah. You could probably fit a good number of tubes in a centrifuge.
They didn't talk about how they were tilted or anything
Mhm.
in there, but they were, and it was a lot of money for undergraduates.
That's awesome. Well, I mean,
Yeah, it's wonderful.
that's awesome. Aw, that's great. Cool. So we're not boiling. We're clearing roots over there. We're
Done about the same time,
centrifuging.
maybe?
Probably, yeah.
Should we
So
do a little break, maybe?
yeah, sure.
Get some snacks.
Yeah.
There's snacks.
Yeah. Snacks and water.
I love these things.
So yeah, does that sound good to everybody?
Yeah.
10 minutes?
Yeah, let's do it.